Hello everyone. Welcome to the new session of photosynthesis in higher plants. So today we are going to solve the NCERT exercise from chapter photosynthesis in higher plants. Okay. So let us start the session with the first question. So here comes the first question. By looking at a plant externally, can you tell whether a plant is C3 or C4? Why and how? So you have to see the plant, any plant externally, and you have to tell that whether it is a C3 type of plant or C4 type of plant, right? How you will do this and why? So this is the question. So here comes the answer. One cannot distinguish whether a plant is C3 or C4, right? So we cannot distinguish the plant whether it is a C3 plant or a C4 plant, right? Just by observing its leaves and other morphological features externally. So by external feature, by looking at the external feature or morphological features, we cannot tell whether the plant is C3 or C4. We have to see the internal structure, right? Internal structure of the leaf or the plant to just deduce that whether the plant is C3 or C4 type. Unlike C3 plants, the leaves of C4 plants have a special anatomy called Kranz anatomy, and this difference can only be observed at the cellular level. So, in C3 plants, we do not have any such type of anatomy, right? But in the case of C4 plant, when you see the internal structure, when you see the anatomical structure of the C4 plant, you see the Kranz anatomy, right? A Kranz anatomy that will be like presence of the bundle sheath cells, right? And this difference can only be observed at the cellular level. So when you see the cellular structure of C3 plant or C4 plant, then only you are going to distinguish whether the plant is C3 and C4. For example, although wheat and maize are grasses, wheat is a C3 plant while maize is a C4 plant, right? So this is one of the difference here. You can see both the plants are the grasses, right? They are the grasses. Like wheat is also a grass type of plant and the maize is also the grass type of plant. But wheat is a C3 plant. Wheat is a C3 plant, whereas the maize is a C4 plant. Maize is a C4 plant, right? So just by looking the external morphology, we cannot come to the conclusion that the plant is C3 or C4, right? We have to see the internal structure, we have to see the anatomical structure, we have to see the cellular level of the structure of the plant, internal plant, right? To come to the conclusion that whether the plant is C3 or C4 type, right? Okay, so moving to the next question. By looking at which internal structure of the plant can you tell whether the plant is C3 or C4? So if you are looking at the internal structure of any plant, which parts of the internal structure will tell you whether the plant is C3 or C4, right? So the leaves of the C4 plants have a special anatomy that is called as Kranz anatomy. We all know that the C4 plant have a very special type of anatomical structures that is also called as a Kranz anatomy, and this makes them different from the C3 plant, right? So C4 plant is quite different from the C3 plant on the basis that they are having bundle sheath cell. This is very, very important. This makes them different from the C3 plant. Special cells known as bundle sheath cells surround the vascular bundle, right? So surrounding the vascular bundle, they are having a sheath, or you can say a bundle sheath like of cells, which is surrounding the vascular bundle, and they are thick walled and have no intercellular spaces. Very, very important point. Okay, no intercellular spaces are present in between the sheath cells, and they are also very thick walled. They are also impervious to the gaseous exchange, so there is no gaseous exchange can take place between this bundle sheet cell. Okay, no gaseous exchange take place from here. So all these anatomical feature help and prevent photorespiration. This is the thing, that this is the advantage of this type of anatomy, that is the Kranz anatomy, right? That it can prevent the photorespiration in C4 plant, whereas in the case of C3 plant, in where such type of anatomical structures are not found or not present, right? But in the case of C4 plant, a special feature, that is the presence of bundle sheet cells surrounding the vascular bundle, so it do not allow the oxygen to reach to the chloroplast of the bundle sheet, right? Because it is having a very thick wall, no intercellular spaces, so it do not allow the oxygen to reach to the the chloroplast of the bundle sheet cell, right, in which the C4 cycle is taking place. So all these anatomical features help and prevent photorespiration in the C4 plants, thereby increasing their ability to photosynthesize, right? And that is why the C4 plant is having the great ability to do the process of photosynthesis, right? So this particular thing is very, very important, bundle sheet cell that is surrounding the vascular bundle. So this was the question actually. By looking at which internal structure of a plant can you tell whether the plant is C3 and C4? So it is a bundle sheet cell that is surrounding the vascular bundle, right? And that is allowing this type of photosynthesis, that is the C4 type of, and do not allow the gases, oxygen gas to reach to the chloroplast region, right? Okay. Moving to our next question. Even though a very few cells in a C4 plant carry out biosynthesis Calvin pathway, yet they are highly productive. Can you discuss why, right? Even though a very few cells in C4 plant carry out biosynthetic Calvin pathway, right? Very few cells are taking this Calvin cycle, right? They are involved in the Calvin cycle, but the number is very, very few in the case of C4 plant. Now you have to tell, still they are very highly productive. Now how can we say that? The productivity of a plant is measured by the rate at which it photosynthesizes. Okay, we can tell the plant is very, very productive by measuring the rate of photosynthesis. How, at which rate the photosynthesis process is taking place in the case of plant, right? The amount of carbon dioxide present in plant is directly proportional to the rate of photosynthesis. Okay, how much carbon dioxide is present in the plant, right? It is directly proportional to the rate of photosynthesis. So if the number of carbon dioxide, the amount of carbon dioxide present in a plant is more than the photosynthesis rate will be more, right? The rate of photosynthesis will be high. So the amount of carbon dioxide present in the plant is directly proportional to the rate of photosynthesis. Now in the same way, C4 plant have a mechanism of increasing the concentration of carbon dioxide. Okay, so C4 plant is having a mechanism to increase the concentration of carbon dioxide, right? Concentration of carbon dioxide, and therefore it increases the rate of photosynthesis. Now in C4 plant, the Calvin cycle occur in the bundle sheet cell. Very, very important. The Calvin cycle is taking place in the bundle sheet cells only, right? It is having the spacious difference, right? So in C4 plant, the Calvin cycle occur in the bundle sheet cell, and the C4 compound, that is the malic acid, from the mesophyll cell is broken down into the bundle sheath cell. So the C4 compound, this is the C4 compound which is formed in the mesophyll cell, and that is called as a malic acid. And for this reason only, this is called as C4 cycle, because the first you can say the stable compound is formed in this type of plant is the 4 carbon compound, that is the malic acid, right? So the C4 compound malic acid from the mesophyll cell is broken down in the bundle sheath cell, and as a result CO2 is released. When this malic acid is broken down in the bundle sheath cell, right, it is releasing the CO2. Now this increase in the CO2 ensures that the enzyme Rubisco does not act as an oxygenase, because there is no oxygen here. There is only one gas, that is the carbon dioxide, which is produced by the broken down of the malic acid. When the malic acid is broken down, it releases the carbon dioxide. So this carbon dioxide that is released is taken by the Rubisco enzyme, right? And it is taking the carboxylase enzyme for its synthesis of the food. So this prevents the photorespiration, because oxygen is not here, so photorespiration process is not taking place. So this prevents the photorespiration and increases the rate of photosynthesis. It increases the rate of photosynthesis. So that is why no photorespiration is taking place in the C4 cycle. So whatever the carbon dioxide is there, it is utilized for the food production. It is utilized for that is why it gives the high production. The C4 plant is highly productive. Okay. So moving to our next question. The next question is, Rubisco is an enzyme that acts both as a carboxylase and oxygenase. Now this is a very, very important enzyme that take part in the process of photosynthesis, that is the Rubisco. It is an enzyme that acts both as a carboxylase and oxygenase. It is taking part as a both carboxylase and oxygenase. And why do you think the Rubisco carries out more carboxylation in the C4 plant? Why it happened that in the C4 plant only the carboxylation is active, not the oxygenation part of this enzyme. So let us see the answer. The enzyme Rubisco is absent from the mesophyll cells of the C4 plant. Okay, Rubisco is not present in the mesophyll cell. Rubisco is present in the bundle sheath cell only. So the food production is taking place in the bundle sheath cell only. So it is present in the bundle sheath cell surrounding the vascular bundle. This bundle sheath cell is surrounding the vascular bundle, and in C4 plant the Calvin cycle occur in the bundle sheath cell, right? So the Calvin cycle is taking place in the bundle sheath cell, not in the mesophyll cell. Whereas in the case of C3 plant, right, the Calvin cycle is taking place in the mesophyll cell. So it is showing both the carboxylase and oxygenase activity. So sometimes there is a process of photorespiration, and that time the enzyme is not available for the carboxylation process, right? So it cannot fix the carbon dioxide at that time. But here what happened in the case of C4 plant, the bundle sheath cell is only showing it carboxylase activity, because this malic acid which is synthesized in the mesophyll cell is passed to the bundle sheath cell. It is not allowing the gases to move, any gas, that is the carbon dioxide or the oxygen. It is not allowing the carbon dioxide or oxygen gas to move into the bundle sheath cell. But the malic acid can enter in this bundle sheath cell. So there, this malic acid is broken down and it releases the carbon dioxide. So the primary CO2 acceptor in the mesophyll cell is the phosphoenol pyruvate, right? A 3-carbon compound. So this phosphoenol pyruvate is present in the mesophyll cell and it is a 3-carbon compound. So it is a primary acceptor. Okay, this is the primary acceptor that accept the CO2 and it is converted into the 4-carbon compound oxaloacetic acid. Very, very important. The first stable compound is the oxaloacetic acid. Now this oxaloacetic acid is further converted into malic acid, right? This is converted into malic acid. So this is very, very important point. Why this is called as oxaloacetic acid. This plant is called as C4 plant because the first stable compound is the oxaloacetic acid and that is why it is called as a C4 cycle. Further, when the oxaloacetic acid is formed in the mesophyll cell, right? It is converted into malic acid and malic acid is transported to the bundle sheath cell, right? So this malic acid which is formed in the mesophyll cell is transported to the bundle sheath cell where it undergoes decarboxylation and CO2 fixation occur by the Calvin cycle. So here the decarboxylation and the CO2 is released and that released CO2 can get entered to the Calvin cycle. Now here no oxygen is available for the Rubisco enzyme. So whatever the carbon dioxide is there, it is utilized for the process of photosynthesis. It is not showing any oxygenase activity. Whereas in the case of C3 plant, it can show the oxygenase as well as the carboxylase activity. But here in the case of C4 plant, only it is showing the carboxylase activity. So this is very important point. And this was the question. Rubisco is an enzyme that acts as both carboxylase and oxygenase, right? But in the case of C4 plant, only the carboxylation process is taking place because the wall of this bundle sheet is very, very thick and it do not allow the oxygen or carbon dioxide to reach there. So what is transported in the bundle sheath cell? That is the oxaloacetic acid is converted to malic acid and malic acid is transported into the bundle sheath cell. So there it is broken down to the carbon dioxide. Okay. Moving to our next question. Suppose there were plants that had a high concentration of chlorophyll B but lacked chlorophyll A. Would it carry out photosynthesis? Then why do the plants have the chlorophyll B and other accessory pigments? So there are the two questions over here. So let us see the answer. Chlorophyll A molecule act as an antenna molecule. Okay, this is the antenna molecule and they get excited by absorbing light and emit electrons during the cyclic and non-cyclic photophosphorylation. So we are having the two type of photophosphorylation. The one is that is called as cyclic, right? And the another is the non-cyclic photophosphorylation. So the non-cyclic photophosphorylation include both the photosystem 1 and photosystem 2, whereas the cyclic one that only contain the photosystem 2, right? So they form the reaction center for both the photosystem first and second. Okay, in the center there is a chlorophyll A molecule for both the photosystem, right? Whether it is a photosystem 1 or photosystem 2, the reaction center is made of chlorophyll A molecule, right? And chlorophyll B and other photosynthetic pigment act as and such as carotenoids and xanthophyll act as a accessory pigment. These are the accessory pigment that is they are presented surrounding of the chlorophyll A molecule. In the center of the photosystem, there is a chlorophyll A molecule, whereas chlorophyll B and other pigments like carotenoids and xanthophyll, they are present surrounding the chlorophyll A molecule. So their role is to absorb the energy. the chlorophyll B and this carotenoids and xanthophyll, they are absorbing the light energy, right? They are absorbing the light energy and transfer it to the center, that is the chlorophyll A, right? So the carotenoids and xanthophyll also protect the chlorophyll molecule from being get photooxidized, right? So this is also protecting the photooxidation of the pigment chlorophyll A. So therefore chlorophyll A is essential for photosynthesis. This is a very, very important point. Chlorophyll A molecule is very important, and that is the core that is present in the core center of the photosystem, whether it is a photosystem 1 or photosystem 2. So if any plant were to lack chlorophyll A and contain the high concentration of chlorophyll B, then this plant would not undergo photosynthesis, right? Because the photosynthesis is absorbing the light. Okay, it get excited, but after the excitation, it transfer electron where? It cannot transfer electron direct to the first acceptor molecule that is coming out from the electron when come out of from the photosystem 1 or photosystem 2. It cannot send directly to that. Only the chlorophyll A molecule can send direct to the pheophytene, right? Not the chlorophyll B. So how much amount of the chlorophyll B is there, right? The concentration may be very high, but still it cannot participate or it cannot undergo the process of photosynthesis. So this is very important point. Chlorophyll A is the main pigment that is present in the center of the photosystem 1 and photosystem 2, right? Okay. Moving to our next question. Why is the color of a leaf kept in the dark frequently yellow or pale green? Which pigment do you think is more stable? Right. So since leaves require light to perform the photosynthesis, okay, for the process of photosynthesis, green leaves require the light. The color of the leaf kept in the dark changes from a darker to the lighter shade of green. So when you keep this leaves in the darker region, the light is not approaching to there directly. Okay. So what you see changes after some days, you see that it becoming lighter shade of the green. The leaves which were earlier darker in shade, now they are changing to the lighter shade of the green. So sometimes it is also turns and yellow, and even you can see the leaves are turning to the yellow color, right? So the production of the chlorophyll pigment essential for the photosynthesis is directly proportional to the amount of light available. So how much amount of the light is available? According to that, chlorophyll pigments, okay, it gets synthesized in the plant. So the light is reduced this time when you are keeping it into this plant in the darker region. So no light is there. So the new synthesis of the chlorophyll will be stopped. And in the absence of light, the production of the chlorophyll A molecule stops and they get broken slowly. So earlier, which this chlorophyll A were present, it will be broken slowly, and also new chlorophyll A will not be get synthesized. So this changes the color of leaf gradually to light green in color and sometime to the yellow also. Right? So during this process, the xanthophyll and carotenoid pigments become predominant. Right? So these two pigments become predominant, that is the xanthophyll and carotenoid pigment, and causing the leaf to become yellow. So basically they are somewhat a yellow pigments. So that makes the leaves a bit of yellowish in color. So these pigments are more stable as the light is not essential for their production, and these pigments are not directly proportional to the light that is received by the green leaves. Okay, so these pigments are more stable as the light is not required for their production. So they are always present in the plant. So this xanthophylline carotenoids are always present in the plants as well. After that, next question comes here. Look at the leaves of the same plant on the shady side and compare it with the leaves on the sunny side. Or compare the potted plants kept in the sunlight with those in the shade. Which of them has leaves that are darker green, and why? Okay. So light is the limiting factor of the photosynthesis. It is a limiting factor because it affects the process of photosynthesis, right? The factors that affect the process of photosynthesis is called as the limiting factor for photosynthesis. So leaves get lesser light for photosynthesis when they are in shade. When they are in the shady region, they get lesser amount of light, and therefore the leaves or plants in the shade perform lesser photosynthesis as compared to the leaves or plants kept in the sunlight. So in order to increase the rate of photosynthesis, the leaves present in the shade have more chlorophyll pigments, right? So to increase the rate of photosynthesis, what they do, the leaves present in the shade, they develop more chlorophyll pigments, and this increases the chlorophyll content, increases the amount of light absorbed by the light, which in turn increases the rate of photosynthesis. Therefore, the leaves or plants in the shade are greener than the leaves or plants that in the sun. Okay. Okay, so here comes the figure-based question. Figure 13.10 shows the effect of light on the rate of photosynthesis. So here we are going to discuss about the rate of photosynthesis. Based on the graph, answer the following question. At which point A, B, C in the curve is light a limiting factor? A, B, C. Okay, which of the part is showing the limiting factor? So first, generally the light is not a limiting factor. Okay, light is not the limiting factor. It becomes a limiting factor for the plant growing in the shade or under the tree canopies. Okay, so only for those plants that are not getting the light directly, right, that are growing under the shade or under the tree canopies. So in the given graph, the light is a limiting factor at the point where the photosynthesis is minimum. So where you can see the rate of photosynthesis is minimum, right? So the least value of the photosynthesis is the region A. Okay, this region is the A, where the rate of photosynthesis is least. Hence, the light is a limiting factor in this particular region, right? Next question was, what could be the limiting factors in the region A? In the region A, what could be the limiting factor? So again, the answer is given here. The light is a limiting factor in the region A. Water, temperature, and the concentration of the carbon dioxide could also be the limiting factor. The rate of photosynthesis is also dependent upon the amount of carbon dioxide that it is having, the concentration of carbon dioxide, the temperature, the water availability, right? So that is also the limiting factor. Next question is, what do C and D represents on the curve? C and D represent on the curve. So point D represent the optimum point and gives the light intensity at which the maximum photosynthesis is recorded. So this is the particular D is representing the optimum point. This is the point at which the maximum rate of photosynthesis is observed or recorded. So that is the light intensity. The rate of photosynthesis remain constant after this point, even though the intensity of the light increases in the region C, right? So the rate of photosynthesis remain constant at this particular point, even though you increase the intensity of light at the region C. So the last question is here. What is the last question? Give comparison between the following. We have to compare C3 and C4 plant, cyclic and non-cyclic photophosphorylation, and anatomy of the leaf C3 and C4. So let us see the difference over here. So C3 and C4 pathway. Okay. So the first difference is the primary acceptor of CO2 is RUBP, ribulose bisphosphate, is a 5-carbon compound in the C3 pathway. That is the Calvin cycle, right? So the first acceptor of CO2 is the RUBP, and that is a 5-carbon compound. And in the C4 pathway, the primary acceptor of CO2 is a phosphoenol pyruvate, and it is a 3-carbon compound. It is a 3-carbon compound, and later it forms the 4-carbon compound, oxaloacetic acid, which later get converted to malic acid in the bundle sheet cell. The first stable product is the 3-phosphoglycerate. So here the first stable compound is 3-carbon compound, and that is quite stable. So it is a 3-phosphoglycerate. In the C4 pathway, the first stable product is the oxaloacetic acid, which is the 4-carbon compound. And this is the reason it is because of the 3-phosphoglycerate is the first stable carbon compound, right? So it is called as C3 pathway because it is the 3-phosphoglycerate 3-carbon compound. And in the same way, oxaloacetic acid is the 4-carbon compound, and it is again in the case of C4 cycle, it is the stable compound, that is the 4-carbon compound, and that is why it is called as C4 pathway. So it occur only in the mesophyll cells of the leaves. Okay, C3 pathway is occurring only in the mesophyll cells of the leaf, right? And it occur in the mesophyll cell and the bundle sheath cells of the leaves, right? So two regions are there. Half of the region, that is the mesophyll cell, is also involved, and the bundle sheath cell is also involved in the case of C4 pathway. Whereas in the C3 pathway, only the mesophyll cells of the leaves are involved for the photosynthesis. Fourth point, it is a slower process of carbon fixation, and photorespiratory losses are high, right? So photorespiration is also occurring along with the Calvin cycle. So the carbon dioxide does not get fully fixed over here, right? Whereas in the case of C4 pathway, it is a faster process of the carbon fixation, and photorespiratory losses are low. So here the photorespiratory losses are quite low. Cyclic and non-cyclic phosphorylation. Next difference is the cyclic and non-cyclic photophosphorylation. So cyclic photophosphorylation, it occur only in the photosystem 1, whereas non-cyclic photophosphorylation, it involves two photosystems, that is photosystem 1 and photosystem 2, both the system, right? Next difference, it involves only the synthesis of ATP. So in the case of cyclic photophosphorylation, NADPH2 is not formed. Only the ATP synthesis takes place. Whereas in the case of non-cyclic photophosphorylation, along with the ATP, NADPH2 is also formed. Okay. Next, in this process, photolysis of water does not occur. Okay, photolysis of water is very, very important step. So in the case of the cyclic photophosphorylation, no photolysis of water takes place. Okay, whereas in the case of non-cyclic photophosphorylation, in this process, photolysis of water takes place and oxygen gas is liberated. So oxygen gas is liberated in the case of this photophosphorylation. Next difference, in this process, electrons moves in a closed circle. Here the electron is moving cyclically, right? So that is, it is moving in a closed circle, cyclic photophosphorylation. Whereas in the case of non-cyclic photophosphorylation, in this process, electrons do not move in a circular form. And here comes the last difference, anatomy of the leaves in the C3 and C4 plants. So C3 leaves, bundle sheath cells are absent. So in this case, bundle sheath cells are not present. Bundle sheath cells are absent, right? In the case of C4 leaves, bundle sheath cells are present surrounding the vascular bundle. Okay, so they are not having any intercellular spaces and the cell wall is quite thick in the case of bundle sheath cell. Next difference, Rubisco is present in the mesophyll cell. Rubisco enzyme is present or confined to the mesophyll cell, whereas Rubisco is not present in the mesophyll cells in the case of C4 leaves. It is present in the bundle sheath cells. This is the big difference in the case of C3 and C4 plants, right? Next difference, the first stable compound produced in the is the 3-phosphoglycerate, is a 3-carbon compound. So in the case of C3 leaves, the first stable compound is the 3-phosphoglycerate and it is a 3-carbon compound. Whereas the first stable compound produced here is the oxaloacetic acid, 4-carbon compound, oxaloacetic acid. Okay, so and that is why it got its name, C3 leaves, C4 leaves, right? Or C3 plant or C4 plants. Okay. Next difference is the photorespiration occur in the mesophyll cells. So photorespiration is taking place here in the case of C3 plants. Whereas in the case of C4 plant, photorespiration does not occur. Photorespiration does not take place. Okay. So this is the difference between the C3 and C4 plants. So along with this, we have completed the NCERT exercise. We have discussed about the solution of the question that is present in the NCERT exercise of the chapter photosynthesis in higher plants. So thank you very much for attending today's session. We will meet with a new session. Till then, thank you.